First of all, I'd like to say welcome to our seminar today. Thank you for joining us. We're very excited to have you. And a special welcome to those of you who are joining us from outside of the Institute, outside of the regular seminar course. Thank you for coming as well. Just as a quick reminder to everybody, if you haven't been to one of our seminars before, for the Q&A, we usually do that at the end of the talk, and you are welcome to put questions in the chat throughout the whole talk, and I will read them at the end. Or if you'd like to ask your question in person, you can use the raise hand function in the attendees tab, and I will call on you at the end to ask your question. As usual, if you are in the course, Paul, my colleague, will be putting the link to the attendance questions.
I think we're good. Yeah, let's keep going.
Sorry, mute that really quick. So yes, if you are in the course, be sure to answer the attendance questions throughout. And with that, I think we are going to go ahead and get started. So it is my real pleasure today to introduce our speaker, who is Dr. Paul Yeager, who is a professor in the Department of Bioengineering at UW with adjunct appointments in chemistry, global health, and oral biology. He's also been involved with the MolES Institute over the years, so we're very happy to have him joining us today. He received his AB in biochemistry from Princeton in 1975 and a PhD in chemistry from the University of Oregon in 1980, specializing in vibrational spectroscopy of biomolecules. After an NRC fellowship at the Naval Research Lab, he joined the NRL staff. He moved to the Center, now Department of Bioengineering at UW as an associate professor in 1987, advancing to professor in 1995. And he's served as the chair of the department from 2007 to 2013. He has co-authored 160 refereed publications, has 41 issued patents, and has co-founded 2 companies, Micronics and is it UBDX? Hopefully I'm pronouncing that correctly. So with that, let's give a big, warm virtual welcome to Dr. Paul Yeager.
Well, thank you. Make sure you can hear me, and then I'm going to try to share screen, and there should be time for questions at the end. So I'm looking forward to this. Okay, welcome. Let's see if I can share the right screen. Let's see. Okay, can you see me?
Yes, that looks good.
You can see the screen, very good. Let me try to shrink this down. Okay, so first of all, the topic is home diagnostic testing before, during, and after the era of COVID-19. Obviously, most of our lives here have been before COVID-19, but there have been things that have changed. And one of them is what it's like to be stuck at home, what it's like not to think that you want to go out to a doctor's office and deal with health and things of that sort. What can you do on your own? What should you do on your own? And how can the medical system change? So first of all, I want to point out that yes, I'm all those things and more, and more is part of the warning. I'm also the chief scientific officer of, yes, a startup company called UBDX for Ubiquitous Diagnostics. We hope to be that. We are sort of a pre-funding company, but we're in the midst of fundraising right now. And I hope I can share with you some of the excitement we've got in that. Where we see UBDX fitting into the world is certainly lab testing has been around forever, and go down to the hospital and you find a whole bunch of it. There have been point of care testing where you have things in a doctor's office, maybe a pharmacy, and there have been home sampling tests where you may spit into a tube or take a swab or a poop sample for that matter and mail it off to some laboratory where they do a test and you get an answer back in a couple of weeks. But with the exception of home pregnancy testing, one test for HIV, and obviously blood glucose testing, virtually nothing's been done in the home until a lot of things got added this last year. And it's because of what happened in COVID-19 and what will continue to happen. So we see UBDX as being a center for lots of different types of home testing. Now, as it turns out, the first one we're focusing on right now is related to COVID-19. So that'll be the follow through here. But understand that the long-term vision, as you see toward the end, is really much broader than that. First of all, I want to thank my lab group. It's small and tight, and here's a picture of us doing things we shouldn't be doing, which is being relatively close to each other, although by now almost all of us are fully vaccinated, so we're getting in pretty good shape. In particular, I want to thank Kevin Jang, Kamal Shah, who actually got his PhD in my lab and is now working, sort of moonlighting with us. Aaron Heininger, Shichu Huang, Sujatha Kumar, Stephen Bennett, Layla Jamal, and a few others who've been working with us. We have lots of partners we're working with around campus. We've recently gotten support from, well, many recent sources, a lot of money over the years from DARPA, WRF, lately money from something called the Emergent Ventures Fast Grant Fund, which gave us a very generous gift, which we've been able to use to take forward some of the COVID-19 work. Is there a need for a respiratory panel test at home? And I'd say the answer is yeah, for a lot of good reasons. Let's see why I am not getting anything happening on my screen. There we go. First of all, determine if there is an ongoing respiratory infection, what pathogen might be responsible, and viral pathogens are certainly interesting. You know, there's what we like to do is SARS, coronavirus 2, the wild type, and a couple, maybe 3 of common circulating variants. That's becoming a hotter topic as the days progress. Also, influenza A and B, which, as you know, right now are very, very low. RSV is also quite low, but beginning to come back. And then we need some internal controls to confirm proper sample acquisition. If you're going to do a nasal swab, you want to know for sure that your negative isn't because you didn't adequately swab your nose. So we tend to look at a human gene for that. And what we really want to do is rapidly communicate the results to the patient, yes, but also the healthcare providers, the physicians, and the public health system itself, to allow people who are testing at home to immediately be part of an epidemiological study, and as well to not just do epidemiology for research, but actually to enable people to move resources to where they're needed. Let's say there's an outbreak on South Capitol Hill. You might want to know that. Why there? Why of all places? Or you may discover from monitoring sewage treatment plants that there is an outbreak in Spokane. The question is, where in Spokane? Could you have people from King County Public Health or the state health system immediately send tests home? So within a day, you'd have an answer as to who actually was spreading it. In the pandemic, there's been a lot of progress to put this kinds of tests, antigen tests, antibody tests, and nucleic acid tests in the home. Some really remarkable progress. Most of those folks were companies that already had flu tests in the works that they were thinking they might put out on the market. And when coronavirus came along, they simply repackaged their tests to do coronavirus and got EUA and got them out on the market. Very few of them are actually in the hands of people yet. Things are just in the process and people are ramping up production. But it's been a real change in thinking about it because people didn't want to leave their homes. They didn't really want to go to big testing centers to get tested. They wanted to do it at home. And frankly, the public health system would be happier if people stayed at home. Now, we're hoping that we aren't forever in a COVID-19 situation, but we do think that this sort of change in where we do healthcare. Say again?
Sorry, just for the attendees, if you could please make sure that your microphones are muted, that would be great. Thank you.
Okay, sorry. So what we've been doing in my lab historically is looking for causes of infectious disease, and that includes antibodies as one very common thing that we've looked at for a while, IgM and IgG, but also we've looked at proteins, both DNA and RNA as well. This is just a picture of antibodies and influenza virus, but we've been looking at both of these things for quite some time in the lab. And the focus has been for quite a while now to decentralize where we do the work such that we could use, for example, a device with a cell phone, use the cell phone to gather the data, maybe analyze the data and send it on through the internet to primary care providers, specialists, emergency rooms, if that's what's needed, clinical researchers, and all tied into your actual complete database, because obviously tests mean a lot more when they're in context of your existing health conditions. Do you have a pre-existing condition that might make you more susceptible to a particular infection? Do you have a history of getting this infection? So a single test data point is interesting, but not nearly as useful as it is in context with all of the health information. So our goal is to keep everything in context. We've done things where we have looked at, for example, could we make lateral flow tests more sensitive using optics? By the way, the expertise of my lab is microfluidics on one side, and the other side is I've been doing spectroscopy since I was an undergraduate many years ago, chasing photons around optical tables to do Raman spectroscopy. So I tend to think of everything as an optics problem. So we looked at the question of whether or not we could use quantum dots to make lateral flow tests more sensitive. It wasn't a new idea, but it turns out it works pretty well. And we were able to get sort of 10-fold, 20-fold improvements in sensitivity of conventional lateral flow tests if we used essentially a UV LED and the regular camera from your cell phone. So we've done a lot with cell phones, and things are really, really, really much improved. I'll skip this. Way back in about 2011, after about a year of discussions, we got funded by DARPA to do what we called the MADNAT project, Multiplexable Autonomous Disposable Nucleic Acid Amplification Test. Now, being DARPA, you'd say, well, everything is for the military, but in fact, the program manager was interested in home testing, wanted to provide or create the technology and then commercialize home tests that could be used by people under prescription from their doctors or self-testing to get complete laboratory data. And we put together a program to do this and worked on it for a little more than 5 years. Lots of important colleagues. Barry Lutz was closely working on this project with Bing Ferric Fung in lab medicine. PATH, a company called originally Epoch Biosciences, now called the Elitech Group Molecular Diagnostics up in Redmond. Seattle Children's, and we had a close partner in General Electric's Global Research Center in upstate New York. They were in it for a while, and then GE kind of got out of the business of thinking about diagnostics. They were in for about 4 of the years of the grant. The aim was all the reagents stored dry, except for buffers if we needed them, and we did. Ideally 1 year room temperature storage, and we wanted storage up to 40 degrees C because not everybody's room temperature is 22 degrees C. We wanted to avoid pumps, so we used paper-based capillary filling when possible. We wanted no user activity after sample insertion. We wanted to be operable by untrained individuals or people that were trained by watching a video on their phones. Simple readout, and in this particular case, no permanent instrument whatsoever. That was a critical initial thing for DARPA. Why? Because everyone knew that there were a lot of people doing what are called point of care instruments, which basically means a box that would sit on a counter in a laboratory. So we decided to take a slightly different route. We were doing several things. First, sample collection and then sample processing. Our initial targets were Staph aureus by DNA, RSV or respiratory syncytial virus, which is an RNA virus. We also did Zika, which was a blood sample, chlamydia and gonorrhea, which are DNA samples. Once we did that on the device, we lysed the sample. These are fragmenting the cells so the DNA and RNA were free to move from the cells, leaving much of the cell debris behind, but most of it was simply caught on the porous materials we're going to use. So the lysis had to be on device. We then did isothermal amplification, and to make as many copies as we could. Typically, you could go from, let's say, 1,000 copies up to 10 to the 12th copies fairly easily, at which point we use lateral flow strip detection as a way to detect things either by eye or ultimately by a camera on a cell phone. The Elitech's isothermal ISDA strand displacement amplification actually was very good. This is an example of real-time detection using ISDA. This happens to be, I believe, Staph aureus. Here's 500 copies. This is about 7 minutes liftoff time. Here is 10 copies. So this is really good, and those are real curves. That's real-time detection by fluorescence in the machine. We also found that using a technique that we co-developed with them, you could also see this in lateral flow strips very easily down to single copies where you get to the point where you've got stochastic results. So in 30 minutes, you've got solid results down to 2 copies. So we said, well, this is it. This is what it actually looked like on the final device itself, where we could see different types of detection. The only major problem with this ISDA was it doesn't multiplex well at all. You can do RNA with single pot reactions if you include a reverse transcriptase. But there's also a primer dimer problem. And the primer dimers come up, and if you had primer dimers, if you were doing things like standard detection with, detecting all the DNA, you would get a strong signal no matter whether you put in a target or not. So we couldn't use conventional intercalating dyes just to light up when you made enough DNA. So in fact, ELITech had developed a lot of sequence-specific probes that allowed you to use this assay very effectively in tube. And our job was to move this into paper into the point of care device. Now, we actually made this thing. This is what it looked like. There's a quarter per scale. It's actually at the same scale. It's just moved pictures around here so you could see it. Sort of 2 packs of cigarettes stacking one on top of the other. I guess I'm dating myself by comparing things to packs of cigarettes. But when we finally finished it, GE had pulled out, so we didn't have a commercial partner that was ready to work with us on it. And the questions were, well, how long does it take? Why does it have to be so big? And how much does it cost? Well, we worked on that because this included not only syringes for fluid delivery, dry reagents, multiple printed circuit boards, 2 AA batteries to run it. So we said, what if we give up on the autonomous part? Okay, we had done a lot to put the batteries in and make sure it could operate on its own. There was enough power from 2 AA batteries to make it work quite effectively. But we said, what if we give up on that? Could we just use USB power? Because everybody's got a USB charger somewhere for their phone. They're, if not completely free, they're virtually free to use USB power. Could we run it on that? And the answer was yes. And we worked on that. So if you want to see the publication, we published on 2016 about this device. Works fine, but the general idea was, well, why does it have to take as long as 55 minutes, which was the total time from sample in to result out? So we said, let's be faster, and let's be smaller, and let's be simpler to the extent possible. So the original idea was the MADAT, which we ran till 2017, simple for end users. It was, however, a bit expensive. We figured that there was about $50 worth of parts in here if you bought them retail. It would never cost that at wholesale, but it was a little pricey. So we said, if we can use USB power for the 2 years from about 2018 to 2020, we rethought this and said, what if we plugged it into USB power and we shrank this thing down, moved everything onto a printed circuit board, and it could be smaller and lighter. And in theory, it could be, fairly simple to use, faster, but still a little expensive because of all the stuff that would be inside there. And in the last incarnation, since we started dealing with product development people who started talking with us, we realized that the original argument to put everything on the device wasn't really necessary. So today's version, which we're just beginning to work on in detail, actually has something like this, but essentially we've thrown all the electronics into a thing we're calling a dock, okay, or a hub, which actually has a power cord, which will plug into the wall, or it could plug into batteries. But with multiple cartridges, each of which does a single test set, or rather one sample, but multiplex tests on it. The advantage of that is you keep all the nice features about the previous devices, but you actually drop the cost per test substantially by putting all the electronics in the permanent component. So it's changed a little bit over the years in terms of what we're going after. And now I'm going to sort of show you in a reasonably short period of time what our progress looks like. One of the key things we did was Mike Roller in our lab, formerly Mike Perfield, developed a really nice valve, which actually uses heat shrink material. We needed a valve on board. And the question was, how can you make it simple? And the nice thing is, you heat this thing to about 75 degrees, you have a slit in a piece of plastic, which you seal. Then you heat it to 70 degrees. And if you hold it right, you clamp it properly, you get a nice round hole and liquids can flow through very easily through it. So that's actually a key component that's filed on out there as a patent. The heating is all controlled by the printed circuit board. Now today, all of the electronics are on the printed circuit board itself, microprocessor, power management, and in fact, a USB interface so we can program this thing. In reality, we wouldn't need to reprogram them because we're going to have all the electronics on the dock. What we will need is heaters, and we're using a combination of surface mount resistors here, and we won't need this two, by the way. And then we have a trace, so basically a serpentine trace. These two are actually monitored with thermistors, so we can hold the temperature to what we need it to be. Isothermal doesn't mean there's no temperature, it means it has to be at a constant temperature. And in fact, most of what we've been using has been at 50 degrees C for the ISDA reaction. And there's the heat, the valve can go up to 70 degrees, that's open loop. To make it faster, the question was, could we use real-time amplification in the glass pad materials that we were using? And I think what you'll see now is this is images of the traces you're seeing in the graph over here, that over, starting at 7 to 10 minutes, you actually see liftoff of the fluorescence of these glass fiber pads. And we said, that's pretty good. This was imaged with a microscope. And we said, whoa, we can do it with a microscope. Can we do it with something simpler like a cell phone? And in fact, Kamal Shah got his PhD doing work demonstrating that we can cut off that lateral flow detection and just do real-time detection in the glass fiber materials, monitoring it with a cell phone. In our initial work, we wanted to monitor two different colors, one of which was an internal amplification control, and the little Texas indicates we're using Texas red for a red dye. Two filters, one over the flash, one over the camera, to allow the cell phone to become a complete fluorescence detection system. All you need is a dark place for it to work. You can get beautiful color images out of these things in fluorescence using this. And in fact, here is a trace of real-time detection on glass fiber of targets and NTCs were the zeros. 1,000 copies or 0 copies on pad. So it works, and the cell phone is an adequate system for doing it. Let's just say that we can further do something that Kamal developed, which was to say, well, if I've got a glass fiber pad and things are glowing over there, where do I look? Obviously, this dark part isn't contributing to the signal. So what we actually do is we look on the paper pad as it's beginning to glow, as amplification starts, find the hottest pixels on there, the ones that are changing the fastest. And you can go from a fairly slow, sluggish amplification to one that actually, looking at the hottest pixels, actually gives you a much faster response time. So we can get earlier results and actually improve the signal to noise ratio of detection. In part because the amplification is occurring in what used to be reagents that were dried on the glass fiber material that have been rehydrated by the lysed sample. By mid-2020, we had this idea for this commercial UBNet project. Again, this is from this Emergent Ventures Fast Grants program and some money from the Washington Research Foundation, designed very much for ease of use by anyone with no lab equipment, single-use disposable, aim for 40 minutes from swab in to result out. Our aim, again, is to use this from paper microfluidics. We want to commercialize an intelligent diagnostic platform, and we founded this company, UBDX, or Ubiquitous Diagnostics, in May of 2020, so almost a year ago. Multiplexing is central to the platform. The idea was we always wanted to do multiple tests, something that would allow you to arrive with a symptom, or for that matter, even to be pre-symptomatic and say, I wonder, since my family has got someone who's sick, am I coming down with something too? Be able to test even before symptoms would arrive. We wanted to do multiple assay configurations paired with something like smartphone technology and optical measurement. Integrating biochemical and digital biomarkers meaning things that you would communicate to your cell phone. This might include data from your Fitbit saying, oh, my heart rate's up. That could mean that I'm about to come down with symptoms of an infectious disease. There's a lot of information out there that we know that's changing the way in which people can use digital data to determine infectious. And there is actually a fairly broad product pipeline for infectious diseases, respiratory, sexual health, et cetera, and a whole bunch of interesting things in chronic diseases. So the concept was, same idea, communication from the UBDX, in this case, UBNAP device specifically into the phone, thence to the company, and then data would move to various places, including hopefully CDC, maybe Jeff Duchin at King County Public Health, hospitals, and then primary care providers. And then the data, the action items could be sent directly back or via UBDX, the company itself. Same idea, swab in for the respiratory panel. Everything would be pretty similar, but we would then report by a cell phone and we would use direct real-time fluorescence. And we've had two methods we've used, the ISDA. We've also taken to using LAMP lately. And why LAMP? Well, because a lot of people use LAMP and it's pretty good at what it does. Again, the experience, swab in, twirl the swab in a buffer. Oh, by the way, the trick was we developed a technique where by unscrewing the cap, you actually compress a blister. The blister then fills a lysis chamber, insert your swab, rotate the swab, break it off, screw the cap back on, which seals the container, at which point everything else happens inside the device. You've got sample diluted, heating the diluted sample to 95 degrees to lyse, cool a sample, then open a valve that flows into the glass fiber amplification zones where your reagents are stored. That rehydrates the ISDA reagents, or let's say amplification reagents. Amplify at 50 degrees for ISDA or 60 for LAMP. And then finally, real-time detection of fluorescence as data is sent to the phone or directly to the company. First thing we had to do was we had to develop an ISD assay for the coronavirus. That actually took us longer than we thought, but it's good. In tube, it's good down to 20 copies in about 20 minutes. Liftoff time, that's at 20 copies. At 2 to the 5th copies, you're lifting off at around 12 minutes. So we said, woo, got an assay. We built devices that include the blister pack and a sample lysis chamber on the PC board. And here's originally 2 zones, and here is a cell phone clamped looking down at an opening in the completed chamber. And this we do on a regular basis. In fact, we're doing this week, again, on more samples. And we did it in a dark room. Obviously, at home, you'd either have to do it in a very dark room or you'd have to build a box around the phone. It works. Early on in July of 2020, we actually were able to see high sensitivity detection in maybe 30 minutes or so, somewhere between 25 and 40 minutes. We could see at 1 million copies into a swab. Okay, and this is 1,000 copies per swab, which we had been which is being told was a reasonably good target. At 35 minutes, we got one pad to light up and not the other one. At 5,000 copies per swab, we're seeing both lighting up, which is at about 270 copies per actual 20 microliter paper swab, and here a glass fiber swab. So our LOD is somewhere around 100 to 200 copies per device, and somewhere between probably 2,000 right now. And that was, on the other hand, that was virus. It was inactivated virus there, but it was in buffer. And then people said, whoa, what about real samples? So we spent a bunch of time, and I'll leave, this is just actual digitized curves from the samples themselves. What about clinical samples? And real nasal swab samples have at least 2 major interference for nucleic acid amplification tests that can slow or kill. One is lots of human DNA to which enzymes bind non-productively. Turns out little sample dilution from about 350 to 500 microliters is plenty for dilution. And RNases are another problem because they chew up any free RNA as soon as it appears outside the viral envelope. And we've solved that by adding RNase inhibitors to the buffer. And in fact, we don't add it to the wet buffer, we actually rehydrate it with the wet buffer. So we're now regularly detecting low copies of viral RNA or inactivated virions spiked into human nasal mucus from healthy volunteers. We've been doing that for a couple of months now, and it's working beautifully. This is just an example in tube of ISDA with, in this case, inactivated SARS virus using the RNase inhibitor. And here's liftoff at 10 minutes. This is 10 to the 4 copies in a tube, but it's really nice and it works quite well. Now, in fact, at first we didn't have all the assays we wanted working. We actually had some trouble coming up with a human gene control assay, and we didn't get a terribly good flu B assay for ISDA. So we said, well, what about LAMP? LAMP has some advantages. It's simpler in the fact that you only have a reverse transcriptase indeed on a polymerase, whereas ISDA requires a nicking enzyme in addition. LAMP allows you to use intercalating dyes. or amplicon-specific probes, but intercalating dyes are classically used, which means it's actually simpler and less expensive as an assay. Furthermore, a lot of LAMP is off patent right now. So actually that's an advantage in certain ways. We've been doing a bunch of dry storage experiments. They're actually, this slide is old. It's no longer ongoing. It's done. We first tried to see, could we do a decent LAMP experiment? So this is, I can't see this, but I believe this is the coronavirus in nasal matrix. And in LAMP, we actually got liftoff at about 15 minutes in tube. We said, well, that's pretty good. And we could actually dry it down and rehydrate it, and it worked quite well. So we said, this is actually promising. So we got ourselves assays for a whole set of assays. We've got, in this case, the SARS coronavirus 2 wild type RSV, influenza A, B, and in this case, human POP7 gene to be the human control. And in tube, at least, we had assays with sensitivities down. The worst one was our flu B assay at 100 copies LOD, but it's 6 copies for POP7, and the coronavirus at 10 copies looked really promising. So we said, whoa, let's see if we can do that. Then the question is multiplex. How do we multiplex? These assays fail if you try to put them all in the same tube. So how do you actually get to multiplex? And the answer is, we said, well, we'll do what we did before. We split into multiplexing. So we created these things we first called lollipop trees. We now use less embarrassing names for them. But the idea is, could we dry down different reagents in different pieces of a single dried pad, which we cut with our laser cutter, pop it into a tray, and then rehydrate all of them from a common channel? And the answer is, do they stay separated? This just shows that, yes, they do stay separated. We've also done assays on them. This is not actual, this is all the same assay, but you can see different size pads are lighting up. Here you can see 6 of them are clearly amplifying. These 2 didn't amplify terribly well, and we're still trying to figure out why the first 2 didn't amplify, but we get pretty good amplification, everyone except the ones that are very close to the entrance point. I'm still not sure why that happened. All 8 of the 15 microliter pads loaded with equal volumes. They obviously not all 15 microliter pads, but they were all loaded with 50 microliters of the master mix. This is, again, cell phone imaging. These were ISDA reactions, and the concept, at least, worked pretty well. We then changed to these, what we're calling quad pads. And why did we do this? Because these are just the right size to fit onto the spiral trace on our printed circuit board. So these actually will fit. So we're actually doing these quad pad multiplexing for most of the work we're doing now. If we change the printed circuit board, we can do more than 4, but this is what we have available to us in the lab right now. You can do 50 degrees C, or you can do 63 degrees for LAMP by controlling the temperature of the printed circuit board, which we have under feedback control. This was imaged again with a conventional cell phone with dual bandpass filters, as we've shown before. This is from February. This is RSV ISDA assays ×4. And again, we've done these. You've sort of seen that. Oh, but this is important. This is data from last week. So this is LAMP. This is RSV, RSV in 1 pad, flu, coronavirus 2, and flu B. So 4 different assays in 4 adjacent pads, each holding about 10 microliters. And here is the progression of a series of images. Here are the quantified images. We didn't normalize these yet, but you can see liftoff for flu A is at around 18 minutes. Here's flu B at around 25 minutes. The yellow one is the coronavirus liftoff at about 31 minutes. And here is RSV, the slowpoke, at around 40 minutes. And this was the first run of the quad pads. So we are rapidly improving on this. We're interested in some interesting issues, which is why so much later than the liftoff and tube. It's a long story, and it's good for a question and answer period. But we're very excited that we've now got multiplexing on pad in a format that will work in our device. And this week, we're actually putting them on the complete black plastic clad printed circuit board device you saw before. So to close, what I really want to do, though, is explain where we're going with this. The idea is this dock or home hub, if you like, might cost $50 at most. Sort of a one-time purchase. It could be purchased by home users. It could be free. depending on whether you buy a subscription to a series of tests or your employer buys it for you or your healthcare company buys it for you. It goes home, at which point you're ready to use cartridges to test. So the first one obviously is this respiratory panel, which will continue to be useful while there's any circulating coronavirus, vaccinated or not, and it will continue to be useful in flu season every year. And that sadly typically comes around when people aren't wearing masks all the time, and we'll probably start having flu very soon again. We're also interested in things that are not just, infectious diseases. We'd include sexually transmitted infections as well, which people want to do in home and usually do fairly quickly. But we're also interested in chronic diseases. There's a lot of interest in inflammatory diseases that result in things like rheumatoid arthritis is a very good one. And there's many of these things that involve inflammatory diseases where tracking how well you're being treated is important. There's a lot of interest these days in kidney disease and whether you can track early stage development of kidney disease markers when you're only in the earliest stages of type 2 diabetes, so that you know when to intervene. And if you are getting interventions, whether the interventions are working. So in certain cases, rapid frequent tracking will actually be very useful and interesting. And this is leaving out, the et cetera, which includes things like cancer and a variety of other, infectious diseases, drug use, things of that sort, all of which could be run from home by reconfiguring the cartridges to do things other than necessarily doing isothermal nucleic acid testing, all of which are easily optically monitored and can be made much more sensitive than conventional lateral flow tests, as we've shown before. Next steps. Right now, we're working on getting some clinical samples to validate them on the alpha in our labs. We want to optimize the performance of assays on the alpha, reduce the total time to result, and actually identify the LOD on device. Validate, as you've seen, the multiplex respiratory panel. Then design for commercialization of this with product development house. And we've identified a really good product development group we want to develop them with, and they're hot to trot, and we're just raising the money right now to get them funded so we can start getting products that we can put into laboratories where people can validate the results in UW labs and elsewhere. And then finally, commercialize the UBNAP as the first one, as the first of the tests coming out of the UBDX configuration. And at that, I will escape and attempt to unshare my screen, which I think, have I unshared yet, or I've been unshared?
Yes, okay. You are good.
And I am open for questions.
Awesome. Thank you so much. That was such a cool talk. That's really exciting. So just a quick reminder to everybody, if you would like to ask a question, you can put them in the chat and I will read them off, or you can use the raise hand function and I will call on you to answer. And I also have one other quick little announcement. As many of you probably noticed, that there was not an attendance question put in the chat, and there will not be one today. So I apologize for hopefully not confusing you on that. But anyway, yes, we can go ahead and start the Q&A. And I guess I have a quick question to get us started off that you kind of answered the first half of it, which was about the distribution of the potential product and the different ways that it could get out. But I think right now a lot of people are really interested about ensuring access to this type of thing within marginalized communities. I was just curious if you'd given any thought to that issue.
Well, it is a very critical question, and we're interested in that. The government is interested in that. The federal government's putting a good deal of money into it. And frankly, a lot of our entry into this particular area was through the Gates Foundation, where the marginalized community is a lot of countries where people don't have nearly as much money to put into healthcare as we do. So that is a reason why reducing the cost per test by taking the electronics off the device made a lot of sense. I worked for 7 years at the Naval Research Lab. The people that brought us the gold-plated screwdriver and things of that sort, the ridiculously expensive devices that met a DoD need. You could design something that would be preposterously expensive and still think it might eventually work for the military. That's not how we're trying to think these days. And for me, the eye-opener was traveling in the developing world, paid by the Gates Foundation, going to several parts of the world I had never visited before and looking at what diagnostics they had. And typically it was paper lateral flow tests, maybe with a plastic housing. where it could be stored at room temperature, when room temperature may have gone up to 40 degrees C for a year and still be useful. So all the criteria that we've put into thinking about both lateral flow protein testing, storing our reagents in dry form and sugar matrices are focused on the question of, okay, I don't need to keep it at -20. Okay, we've had this experience now with the vaccines. There are ways in which you can preserve pretty much any kind of reagents, mostly with a combination of sugar matrices and proper excipients to redissolve things. So you can deal with that problem. The issue of how you get it into the hands of people who don't want to be tested in the first place is a very interesting one. And we have been talking, my CEO partner of mine and I, Kate Sheridan, about the question of how you do get that. And the answer is there are lots of things that grow through basically social networking. You create groups of people who talk to each other, describe their symptoms, describe how they're coping with diseases, and use the power of social networking to actually bring people to be willing to engage in regular healthcare. Because, legislating it into happening doesn't make it work. A critical issue, of course, is simply the financial issue. How do you make it acceptable? And if you look at how much it costs the federal government and this country to deal with the COVID-19 crisis, and that's people filling up hospital systems. Just look at how much UW spent on the hospital system. You can make a very good argument that the government ought to give these away, tests and the doc, because the advantage to the healthcare system for not having that many people getting really seriously ill is there. And that's even assuming we don't have anything more dangerous than the COVID-19 one. When we were, and I'm also, through my global health time, I'm part of the, what we started a few years back, which is now called the Alliance for Pandemic Preparedness, which is ironic because we weren't. Definitely not at all. And we're sort of looking hard at what we can do. And the answer is we want the canaries in the mine shaft. We want sentinels. We want people who are out there who can say, I've got a cough, but my test is negative according to the flu. Do you think it could be something else? At which point we put the genes from a virus that just showed up in Saudi Arabia last week, get it on the devices, rapidly get it out to people by FedEx or, have Amazon deliver it to your door. And 24 hours later, you can be doing a home test without risking sharing your disease with people at the doctor's office. So the ability to isolate people wherever they are at whatever financial level they can do, it's got to be part of it. And again, you have to bring the community into the process and help them to understand that they are doing it for their community, not just for the man in some part of the country or the doctor at the hospital they've never visited before. So yeah, central issue, absolutely. Not just this year and today, but it always has been an issue, and it's a big financial thing. One person gets sick down the block or in your other neighborhood, you're sick too. That's what we've learned.
Thank you.
Sure.
All right, does anyone have a question? I guess I'm also curious, if you guys are formulating questions, another thing that I was wondering about, I think it's so cool that it could have the ability to test for other things too. I was just curious how rapidly you think a new cartridge could be developed for something like if a new pandemic arises.
Sure. Well, a lot of things that are going on right now in places like DARPA are to generate, new vaccines on a dime. The reality is that we got vaccines out very quickly. The advantage of a diagnostic is it doesn't have to be proved safe. It just has to be proven sensitive. So in fact, the minute somebody in a laboratory in the Western Cape laboratory in South Africa picks up a new variant of a virus, the reality is that sequence goes to somebody down the block. And as long as you're able to generate nucleic acid sequences, we have everything in place where we can dry it down on the pads and get them out the door. The argument would be that you should be able to mass produce within a month a new strain. There's no reason that couldn't be done. We're talking to BARDA this week, okay? And BARDA is sort of the kind of organization that's supposed to do these things. Up until last year, BARDA had never stockpiled a diagnostic before. They did a lot of therapeutics, but they never stockpiled diagnostics. What you don't want is a year's worth of last year's diagnostic. What you want to do, and they're actually big on this, is to create the manufacturing capability and then call on it to come into action. So the advantage of having a business that's based on things like chronic diseases, checking seasonal flu, is you have all the stuff in place. People have the devices preloaded, as it were, into their houses. And then all you have to do is remanufacture the right sequences in the disposable. Now, having said all this about nucleic acid tests, they're really sensitive, they're pretty fast. I've been doing immunoassays for 20, 30 years now. And the answer is they're pretty good too. And for certain things, they're better. When you want to detect a protein, when you want to detect a toxin, when you want to look at a metabolite in the body, those things are fine. And there's no reason you can't use the same format, the exact same reader, but with different lights on inside, literally. The plan is to have different filter combinations and light combinations to work in white light, if that's what you need, to do the UV illumination, to do quantum dots, for you get more sensitive lateral flow assays. So the beauty of this optical approach is you can take pretty much any type of clinically relevant assay and use the optical version in here. We're even, discussing things like, INR measurements for people who are on blood thinners. Could you do that? You bet you can. What you have is basically a small microscope sitting inside there. You can do optical measurements. So, like I said, having been an optical jock since I was an undergrad, I see everything as an optics problem. And the beauty is most people who've done clinical chemistry over the years have actually also seen things as optics problems. So this is a nice format for doing that kind of home testing.
Yeah, great. So we have another question in the chat here. This is from Susan Johnston. And she asks, is the current situation that results of tests are propagated to all of these locations/organizations? What about privacy concerns?
Well, the answer is right now, the company is not in business yet. So we're not spreading information anywhere. Clearly, right now, if you do an HIV test at home, which is sort of the scariest of most of them. Pregnancy is also a scary test, but home HIV testing has got a lot of issues. And right now it's not connected to anything. They offer you a hotline, but that data is yours and yours alone if you do it in the privacy of your home. Public health people hate that. They are really hoping that people who get a positive test are going to go in, because what they want to do is they want to do contact tracing. They want to find out who your partners are. They want to be able to put bounds around things. And contact tracing is the only way you actually stop epidemics. To contact tracing, they go, well, your testing isn't really useful. You might conclude that maybe you should stay home, but maybe you don't think so either. Presumably, if you do test, it's because you're inclined to be not just selfish, but also thinking about the herd. But we thought about this. There's an awful lot of thinking going into what the software is going to be around this. And the main idea is that your cell phone will be the primary connection to the device. And one would assume that the first thing you will be able to do is decide who will get your information. Do you keep it to yourself or do you share it? The issue that we're wondering is, would you be able to do all the data analysis on the phone? And we do believe that the phone has got plenty of compute power to take curves or line analysis and perform them on the phone itself. Not a problem. You download the code. The question is, you ought to be able to decide where it goes. If you don't trust the federal government, you can send it to nobody but your personal physician or to no one. So we think that's going to be part of the deal. Now, if your test was sent to you by your employer, okay, you're getting them free because Boeing decides it's going to give everybody flu tests in flu season, there may be different deals there in terms of who gets the information. So it's really going to depend on who's paying. If you're paying, you get to control. If your employer's paying, you probably have to do what the employer wants you to do with the test. So I think it's going to come down to case-by-case circumstances. Students may be required to demonstrate negative test results at a certain time. Will that be a condition of your staying enrolled at the university? I don't know. These are the sorts of legal things we're going to have to start arguing about, and we're all going to be talking to lawyers.
Thank you. All right. Do we have any other questions? Remember, you can either put them in the chat or you can use the raise hand.
Mm-hmm. And I'm on campus and open to questions. If people come up with the questions 15 minutes after this, after we're done, happy to do that. And I will say that my deep connections for a long time when we were developing a flu assay was entirely protein-based. And it was lateral flow testing, and we put devices in Children's Hospital and had nothing to do with nucleic acid testing whatsoever. It's still a good platform for that. I saw there was a question that just came through.
Yes, this is from David Jackson, and he asks, would you please elaborate more on how the glass fiber technology works for readout and how the nanoparticle technology enhances sensitivity?
Sure. For the glass fibers, what we started working on back in about 2008 was replacing a lot of conventional syringe pumps using capillarity as a way to move fluids from point to point. It means that you can get fluid movements under control, sometimes with a valve, but often without valves, just by which piece of paper you wet and then what the timing is. We realized that in conventional lateral flow tests, like consider a pregnancy test, there are pads. One's called a conjugate pad upstream where the urine goes in, you wet detection antibodies that are gold labeled, so they have gold nanoparticles on them, and those then mix with the sample as it flows through the material. The storage component are those glass fiber materials, and they work really well. So what you do is you take a glass fiber pad, you add a solution of, let's say, the master mix for the nucleic acid amplification, and we then freeze dry them. So we lyophilize them, at which point they can be stored dried for up to a year, which is nice. So that keeps all of your reagents in dry storage at room temperature in nearly anhydrous conditions. You put some Dry-Rite in the device to make sure it doesn't get soggy and gooey. So mostly what it's there to do is to allow the reagents to be stored and then to be rehydrated by the sample as the sample wicks into the glass fiber pad. So it's a combination of storage medium as well as a mixer, because the fluid moves in, it covers the entire area because we only fill it to the point where it's 50% solids. So there's room for things to get in there, and then they rehydrate. So that's what the glass fiber material does for us. I'm remembering that the second half of the question was, I'm sorry, what was the second half?
How the nanoparticle technology enhances sensitivity.
It turns out that the conventional lateral flow test that you'll see either uses dyed microparticles, or more commonly 5 to 20 nanometer gold nanoparticles. Gold nanoparticles absorb an enormous amount of light at 530. They look dark red when you look at them, to purple, depending on how they're going. But it turns out that you can only see so many of those on a white background, like the paper in the nitrocellulose paper that's in lateral flow tests. So we calculate that for the typical sort of 1 millimeter wide line, you can't see fewer than about 10 to the 8th of those particles. Well, you've only got 10 to the 6 things you're capturing. You may not easily be able to see them with gold nanoparticles. So people have been trying to figure out ways to make that darker or brighter. One of the ways you can do that is you chemically increase the size of the gold nanoparticles. We've also worked with HRP and horseradish peroxidase and oxidation reactions that produce precipitates of dark materials. But the other thing you can do is use a fluorescent nanoparticle instead of an absorbing nanoparticle. So quantum dots are intensely fluorescent, and you can excite them in the UV. And that leads to maybe 200-fold better sensitivity than you get with just white light detection of gold nanoparticles.
Great. Looks like that answered the question. All right. If nobody has another question, we can go ahead and wrap up here.
Okay.
Thank you again, Dr. Yeager. This was so cool.
Glad to see you guys coming in on the beautiful day. I hope you go outside and enjoy the sunshine now.
Yes, very much so. All right. And thank you, everyone, for attending. And we will see you again next week. Thanks.
You all take care.
Bye.
Bye.